This is an annual event which is a chance for us to really display some of the research that's going on in the department and share it with a wider community, people who are fellow researchers or others who just have an interest in the topic. As I say, the lecture is being broadcast online, but I should also mention it's also being recorded, and eventually it will be available as a recording on our website. If I could just mention also at the end of the lecture, everyone is very welcome to join us for a drink in the common room. So we'll be ending about 5:30 or so, and then we'll go over there between 5:30 and 6. Anyway, it gives me great pleasure to welcome as our speaker this afternoon, Dr. Danny Wilson. Danny is Associate Professor in Data Science here in the department and in charge of a very important and growing part of the department's work in the area of data science. He's also a fellow of St. Cross College, just around the corner from here, and most importantly, in the context of his talk today, he's leading a research group at the Big Data Institute at the Nuffield Department of Population Health. He's been very involved, as you'll see from the title of his talk, in terms of the use of data science in the question of the pandemic and so on. So I'm very much looking forward to hearing what he has to say, and you'll all have a chance to ask questions afterwards. So with no more ado, Danny.
Thanks, Tom. Thanks very much. Thanks for coming today, and welcome also to the people online. I think we need to say that it's being recorded. So it's a pleasure to talk to you today at the Ruley House lecture, and also to meet several of my colleagues in person for the first time, although I've met them previously on Microsoft Teams. The title of my talk and the subjects this afternoon is Identifying COVID-19 Risk Factors and the Role of Data Science in the Pandemic Response. So I'm based at the Big Data Institute, where my research group in infectious disease genomics consists of these friendly faces, some current and recent members of the group, and I'm going to be talking about some of their work today. The Big Data Institute is a biomedical science research centre based on the Old Road campus in Headington in Oxford, fairly close to the centre of town. This is what it looks like on the ground. And if I was to try to encapsulate what the subject of our research is, then it's this. What can genomes, whether they're human genomes or microorganism genomes, what can genomes tell us about infection, about the process of infection, and about risk factors for infection? And ultimately, we could try to use that understanding to reduce infection, suffering, disease, and mortality. We're trying to achieve this aim at different levels. So we're facilitating infection research by collaborating with national cohorts. We're developing computational methods for big data analysis, and we're trying to understand the role of genetics in specific infections such as the bacterium Staphylococcus aureus, a major cause of sepsis worldwide, and viruses like COVID, which I'm going to talk about more. In my role as Director of Studies in Data Science at the Department for Continuing Education, I have oversight for 30-odd courses, including an open access program encompassing weekly courses featuring overview of data science, programming in Python, R, Excel, JavaScript, and statistics for non-specialists. We have weekend courses in such topics as fake news, natural language processing, and quantum computing. And we've recently launched a new asynchronous or Moodle program in artificial intelligence with 4 courses, starting with an introduction to AI and then covering applications of AI, machine learning, and applied statistics. We also have in our courses the number mysteries based on the book by Marcus de Sotoy, the Professor of the Public Understanding of Science here at Oxford. And we have an award-bearing course, which is the Advanced Diploma in IT Systems Analysis and Design, looked after by course director Dr. Seppi Chakave. Which brings me to the question of what is data science? When I first started, my colleague and historian Elizabeth Gemmell challenged me to define data science, and off the cuff, I came up with something like this. It's interpreting information to draw inferences or make predictions about the world, which admittedly is quite broad. So it might be slightly more specifically applied to the natural world, which would mean biology, for instance, which is my background, or the physical world, or maybe the environmental sciences, or even you could say data science is interpreting information to draw inferences or make predictions about the past, which actually sounds quite a lot like the definition of history. So clearly this doesn't encompass exactly what data science is. There are some other flavors, such as it tends to be quantitative in nature, but not exclusively, certainly digital, and usually sources are contemporaneous, but not exclusively. So within this broad subject, I've tried to tease out several strands, and I've tentatively called these informatics, engineering, and inference. So informatics would be the challenge of processing data, of sampling it in the first place, processes known as data cleaning and data wrangling, and trying to make sense of data and apply structure to it, for example, in the design of databases, all of which are designed to make the data accessible and therefore usable for downstream applications. And those downstream applications would encompass the engineering and the inference strands of this. So engineering is the application of data to real world problems. For instance, supposing we are in real time monitoring the vital signs of a patient on an intensive care unit, using data streams from multiple sources and trying to assimilate those in order to raise a warning in case the patient's vital signs decline suddenly. And finally, inference, where we're trying to use data science to understand the world around us. If you like, this is the science of data science, and trying to glean something that we didn't know already about the processes underlying the patterns of data that we see in the world around us. And more specifically, there are subdisciplines, including, very importantly, the sampling of the data in the first place, theoretical methods development, implementation of those methods in algorithms, and then gluing those things together in workflows, processing the data, which also encompasses the software and hardware considerations of dealing with very, very large amounts of data and doing so securely in the context of information governance. And then finally, interpretation of the data and trying to use it in an application to predict or to infer. So focusing on this last of the strands, the inference side of data science, the modern view of inference goes something like this. We start off with quantities of interest, something that we care about, perhaps a question that we want answered, and we have some data, and that data might have been collected specifically for the purpose. It might even be the result of an experiment, but very commonly it's observational data which may actually be being repurposed and exploited opportunistically to answer a question that perhaps it wasn't originally designed to answer. And the key cog in this machine is what's known as the data generating process, a concept which connects the quantities that we're interested in the first place and the observed data. And this is a model. It's very often, probably usually a mathematical model. And the idea is to take the observed data that we have and then work backwards by contextualizing that data through the data generating process in order to learn about the quantities of interest. Okay, so I'm going to talk specifically about the role of data science in the pandemic response. And data science has been ubiquitous in the many fronts on which we've been fighting the pandemic, as I'm sure you're aware, from characterizing the nature of the threat through to making policy, repurposing drugs, developing vaccines, actively surveying the population to monitor the progress of the epidemic and to understand the impacts of interventions, and identifying risk factors which explain why some people are worse affected than others. So that's the one I'm going to focus on, but I want to briefly give you a few examples of how data science has has helped in some of these other areas. I want to start with understanding the nature of the threat. As you'll recall, the pandemic started or was first recognized from an outbreak in the city of Wuhan in China. And this graph from an early paper in 2020 shows in orange cases of what we now call COVID linked to the Huanan seafood wholesale market, and then in pale orange other cases which were not known to be directly linked. And so this is the first illustration of taking pieces of information, including anecdotal observations that there's something strange going on at the seafood market, and putting it together systematically into data and interpreting that data through systems such as contact tracing and looking initially at risk factors and the profile of the sorts of people that were becoming sick. So in late December, the outbreak was initially announced. The market was closed on the 1st of January 2020. And a novel coronavirus was officially announced as the causative agent by the Chinese CDC, the Centers for Disease Control and Surveillance. And 2 days later, on the 10th of January, the China CDC publicly shared the genome sequence of the novel coronavirus for the first time. This was an important moment. The genome of the coronavirus is about 30,000 letters long, consists of A's, C's, G's, and U's. And understanding that sequence was a crucial moment for many of the things that subsequently followed. So the first thing that it allowed scientists to do was to understand taxonomically what kind of virus is it, and therefore what are its likely characteristics. And from a collection of genome sequences, one can build a tree. The analogy with a tree here is that the virus sequences are the leaves in the tree, and as backwards in time they find common ancestors, those are the branching structures of the tree which finally coalesce into a trunk and finally a root in the tree. If I turn this around, what you can see is it contextualizes the genome sequence of SARS-CoV-2 in the bottom right with other closely related viruses, including, of course, SARS, which caused an epidemic in Southeast Asia around 2001. But more closely related than that are viruses found in animals, including bats and even pangolins. This was a cause of considerable concern, discovering that it was a coronavirus, because the case fatality rates of other known coronaviruses have been extremely high, between 10% and 40% for SARS-1 versus MERS. The genome also immediately gave the protein sequences of the proteins encoded by the genome, one of those being the spike protein. And so it was from that initial sequence, which was made available in early January, that those developing vaccines were immediately able to target a specific protein, whether that be the RNA vaccines like Pfizer and Moderna, or the vector-based vaccine such as the Oxford AstraZeneca one. Genomes also gave us the opportunity to look at the relationships between the different cases, and it was clear, although I'm not going to show you, from a tree based on 30 of those sequences that were available from late January, that the diversity amongst the Wuhan outbreak was very, very low, meaning that it was most likely a point source created by a single transfer from an unknown source into the human population, and that probably happened just once. Other things that were apparent very early on at this stage was basic epidemiological characteristics. It affected older people more, and it affected males more. So surveillance was important throughout the pandemic, and to many people, this became a familiar sight. This is the Johns Hopkins University COVID-19 dashboard, and initially, this was put together by PhD students and postdocs and researchers around the world, manually entering data as it came in a drip fashion into a Google spreadsheet. Now, we're now at 500 million cases and rising, and that's not how it's done anymore. But as the surveillance machinery got off the ground, then more epidemiological characteristics became apparent. in early March, the epidemic in the UK was started to take off, and it was becoming apparent that this was it was becoming apparent that the case fatality rate was much lower, more like 1%, so much less severe than SARS-1 and much less than MERS. Other things had become apparent even earlier than that. So perhaps the reason why this was destined to cause a pandemic was that most transmission was either through people who were completely asymptomatic and therefore showing no symptoms at all, or quite unusually pre-symptomatic, so people that got sick, but the transmission occurred before they got sick, and that makes it extremely difficult to put into practice any intervention strategy if you don't know who's sick and therefore who is passing it on. Surveillance continued, and there were several important surveillance arms here in England, including from Public Health England, now UKSA, the UK Health Security Agency, the Office of National Statistics, and UK Biobank, more on which later. And this surveillance fed into policy. Early on, policymakers were talking about flattening the curve, and in this white paper from the Imperial College COVID-19 response team, you can see what they meant by that. So in black is what they predicted from mathematical models would happen if you did nothing. There would be a precipitous rise in cases. The total number of cases in critical care would be extremely high. Hospitals and other key services would be overwhelmed, and there would be knock-on calamitous effects. And then the idea of doing progressively more would flatten the curve in the sense that it would reduce that peak, making it more manageable. But the side effect is therefore to prolong the epidemic in England. And this was the first appearance of the lockdown sawtooth. So in this policy paper, it was suggested that one way of handling the epidemic was to introduce lockdowns to bring cases right down and then have a period of freedom again, followed by lockdowns which were triggered by rises in cases. And effectively, that's what came to pass. The details are maybe not quite the same. The periodicity is perhaps longer, but essentially that's what was happening in a scenario where we had effectively no good treatments and a vaccine was a long way off. Data science has been important for drug repurposing, and I'll give you an illustration of that through the recovery trial, which was masterminded by people at the Big Data Institute, and indeed a network of healthcare workers and biomedical researchers around the country. So the idea of recovery was to conduct clinical trials, very important if you're going to be developing policy about which drugs to give thousands of people. And it was able to demonstrate effectiveness for some treatments and non-effectiveness for others. Notably, dexamethasone was an effective treatment. So here you see what happened was, and this was an experiment, not a natural experiment. It was a randomization in hospitals with real patients. And on day 0, those patients were randomized either to usual care or to usual care plus dexamethasone. And you can see that in the days from the point of randomization, mortality increases, but it does so at a lower rate for those on dexamethasone, demonstrating a strong and statistically significant benefit of this drug for those individuals who needed either mechanical ventilation or oxygen treatment, but no significant benefit for others. This was the first ray of sunshine, really, because it demonstrated a strong effect of a widely available and cheap drug that has saved a lot of lives since then. The flip side of the coin being the widely feted hydroxychloroquine, a malaria drug, which in the trial showed no benefit relative to usual care. And then mid-November came news, which I think was probably better than we could have hoped for, high effectiveness of not just one, but several vaccines which are in development. In particular, the RNA-based vaccines, which are a new technology, showed extremely high effectiveness, and the vector-based vaccine from Oxford and AstraZeneca, probably arguably with its 70% efficacy, was higher than most people were fearing. What I want to focus on is data science and identifying risk factors. So the question here is, why do some people get sick and some people don't? And is it due to identifiable risk factors? And we already knew that there were several important ones: age, sex. These were apparent even from the very earliest days. And then from initial analyses of the epidemic in England, it was clear that non-white ethnicity was a risk factor, having comorbidities, and also a range of socioeconomic factors. The ability to carry out these kinds of studies looking at risk factors in the UK is in large part due to the high participation amongst the British public in biomedical science studies. It's estimated that well over 1 million people in the pandemic years 2020 and 2021 participated in clinical research of one type or another. And although that was almost double the number of the previous year, actually this is part of a growing trend. In the context of genetics, this shows the number of customers in consumer genomics companies over the last 10 years, and it's risen and risen. What happens here is that individuals want to get their genomes characterized using a technique known as genotyping, and that can tell them a number of things, including about their genetic ancestry. It tells them if they have any DNA relatives that have also signed up for the service, and it can also tell them about risk factors to particular diseases, some of those with the strongest and unusually strong predictive power include Parkinson's and some kinds of breast cancer. One of the most remarkable of these cohorts is based in the UK, and it's called UK Biobank, and it has genetic and health data from 500,000 people in the United Kingdom. And the UK Biobank is who we worked with mostly during the pandemic. So UK Biobank was set up around 15 years ago. It may be that some people in the room or on the call are members or know family members who are part of UK Biobank. It was recruiting initially people in middle age and following them for the rest of their lives with access to detailed medical records and a range of other information, including physical measures, genetics, lifestyle, physical activity, cognition, hearing, online surveys, biomarkers, and imaging. And the aim of this longitudinal cohort was to understand risk factors for common diseases of aging. It turns out it was also useful for understanding COVID. So before the pandemic, my group was working with UK Biobank, Chief Scientist Naomi Allen, and colleagues from Public Health England, Anne-Marie O'Connell and David Wiley, to try to improve the information contained in this cohort about infection. And Public Health England has a database, it's called SGSS. It's a microbiology database. Anyone who goes to a hospital or indeed the GP with either a notifiable infection or an infection that requires antimicrobial resistance profiling, those infection events will find their way into SGSS. So it's updated every 24 hours and it collates data from the NHS and Public Health England labs across the country. And what we were seeking to do was to take the English members of the UK Biobank and link this data to their profiles in order to enable researchers around the world to study risk factors for infection. Well, in March of 2020, we discovered that PHE was routing the SARS-CoV-2, the virus that causes COVID-19, routing the SARS-CoV-2 PCR test results into SGSS. And so, of course, immediately we tried to link those data to UK Biobank to facilitate research of COVID-19 in this cohort. And what happened is that people taking these PCR tests, whether they be patients, healthcare workers, or general population, those results get sent to Public Health England in Colindale in London, and then using the algorithms that we had been developing, we put them immediately into practice, and we linked not only to UK Biobank, but also other large cohorts based in England. And we were able to provide weekly updates from late March, and as early as April, you started seeing preprints online reporting results of associations between particular risk factors and COVID, or rather SARS-CoV-2 positive infection status in the UK Biobank cohort. So that's a remarkably short time frame. And with other data sources that Naomi Allen linked to UK Biobank, including on hospitalization and on mortality, then people were able to start to understand in some detail these risk factors in this large cohort. So the question became, well, why do some people get COVID and others do not? And recall that about 80% of people who catch SARS-CoV-2 probably don't show any symptoms. That was based on the original strain. I don't know what the figures are now. And of those who require hospitalization, a total of about 1% of the population, based on initial estimates, may have died. So we wanted not only to contribute to linking the data between Public Health England and UK Biobank, but also to be involved in the analysis of it. And so we conducted analyses of genetic risk factors in the UK Biobank for different health outcomes. And we contributed these to an international collaboration known as the COVID-19 Host Genetics Initiative, which drew on sources from 200 different studies around the world, some of which were retrospective studies, like in UK Biobank, where they're trying to learn about COVID in a cohort that's already established, some being prospective, where they would go out and they would specifically focus on individuals with the most severe forms of COVID and attempt to genotype them and gain other information about their health medical records after the fact. The COVID-19 Host Genetics Initiative put together a phenotyping committee which defined a range of outcomes in order to harmonize analyses across the world. And in doing so, it defined this pyramid of progression to the most severe form of disease. So at the bottom, you have the general population. A proportion of those are laboratory tested, and of those, some proportion are infected with SARS-CoV-2, of whom some are hospitalized with COVID-19, and at the peak of the pyramid are the most severe cases requiring respiratory support, or those who died from COVID. And the idea was to take those top 3 tiers of the pyramid and compare them to the rest of the general population. And so those are the analyses here called A, B, and C. A, the most severe, and C, the least severe. And in all 3 of these analyses, the design is a case control study where the cases are those in the orange top 3 tiers and the controls are the rest of the general population. We have information on their genetics and we have information on their health outcomes. And to give you a sense of where the signal comes from in a genome-wide association study, or a GWAS, here's a cartoon. So every gene has genetic variants. They're often called SNPs, or single nucleotide polymorphisms. And essentially what a SNP is it defines 2 alternative forms of a gene, or 2 different alleles. And the standard form is known as the wild type allele, and the alternative form is often referred to as the mutant allele. In this graph, on the left you have cases with disease, and on the right you have controls without disease. And the individuals with a green dot are those individuals with the mutant allele. And what you can see in this example, toy example, is that the mutant is rare, but it's equally rare in cases and controls. In this row, the mutant is common, but it's equally common in cases and controls. And the third row illustrates the sort of signal that you're looking for, where you have a mutant which is common in cases and rare in controls. So applying this to UK Biobank, along with other groups in the US and Canada, and then meta-analyzing those within the consortium with a large number of other studies, the COVID-19 Host Genetics Initiative reported a large number of genes around the human genome to be associated with the 3 different outcomes that we're investigating, including in blue, infection, in orange, hospitalization, and in grey, the most severe form of COVID. And what you can see is there's a huge signal in chromosome 3 that's specifically associated with hospitalization, or the most severe form of respiratory COVID. And on chromosome 9, there's a strong association with specifically with infection, and that's at the ABO blood group locus. And I think off the top of my head, I think that individuals that are blood group A are at 1.5 times greater risk than those who are blood group O. So this demonstrates that genetics is important, probably more important than most pathogens. And bear in mind that, of course, SARS-CoV-2 is a new infection in humans. When you look at other things such as susceptibility to bacterial infections, you don't see the same kind of strength of signals. And that's generally the case across the board. So very strong signals. And these signals tell us at least two things. The first, and I suppose the shining hope, is that they lead to new treatments. How might they do this? Well, the genes which are found to be involved, often something is known about the biology, including possibly the biological pathways that they're involved with, or indeed other diseases that they might be involved with. And in perhaps the best case scenario, there are already drugs developed which target that pathway for those other diseases. So it's a way of potentially identifying drugs for repurposing. But the second thing that it does is through a greater understanding of the biological pathways underlying COVID, is that it tells us something about the disease process itself. And in particular, it was revealed that there were two distinct mechanisms that predispose to life-threatening COVID. The first of those is difficulty controlling viral replication in the first place inside the body. And the second is a tendency towards inflammation and blood clotting. So interest in risk factors is not solely restricted to an interest in genetic risk factors, and I want to talk to you a little bit about non-genetic risk factors. Starting with those first preprints that appeared in April of 2020, there's been a lot of research into risk factors for susceptibility, hospitalization, and mortality due to COVID in UK Biobank. And my PhD student, Nick Arning, surveyed those papers and looked at their headlines and made a table of the sorts of risk factors that come up often. Can you see, just about see? Okay. So at the top of there, for susceptibility to infection, you have obesity. I don't think that will come as a surprise to anybody. Ethnicity, smoking, cholesterol, medication, different occupations, blood sugar, and cardiovascular disease, to name the top 8. And when you look at the other outcomes, shall I ignore that? Okay. And you see similar risk factors when you look at the other outcomes: hospitalization and mortality, and obesity in particular is reported again and again. But other things come up towards the top, such as mental health, deprivation, lung disease, kidney disease, and so on. So to come back to the data science inference concept, here we have some observed data and quite a lot of it. We have quantities of interest. For instance, is smoking a risk factor for hospitalization from COVID? And what we need to tie these things together and make sense of the data is a data generating process. So let me illustrate some of those points with the following example. Does drinking alcohol increase the risk of COVID-19? The data is whether individuals are hospitalized or not. And there are many potential risk factors which in a linear model might try to explain one from the other. For example, obesity as measured by waist circumference, age, being male, being white, various indices of deprivation, and then the quantity that we're directly interested in, alcohol consumption. And so how does one build a data science model to look at this? Well, I'm going to try to illustrate. The way to do it in this case, because hospitalization is a binary trait, is you can directly define a probability that a particular individual in the cohort was hospitalized due to COVID or was a member of the general population. And typically this kind of analysis is conducted with a class of mathematical models known as linear models. And within these linear models, you add up a bunch of risks and then you transform it to a probability scale using a function known as a logistic function. If you're interested, the formula is in the bottom left corner. So how might this work? There's usually a baseline level of risk in the cohort that you have to account for, and then you want to be including various other risk factors that you know or suspect to be important. So here we have age. So age is a potential risk factor, and it has a coefficient in grey, which is the risk, let's say, per year. So we've measured age in years, and we have an added risk per year, and we add that in to the total risk. So the grey boxes are parameters of the model. Maybe we want to include sex, ethnicity, waist circumference, indices of deprivation. These all go into the model. And then the quantity that we're interested in here is whether alcohol consumption, when frequent, alters risk. And so we really care about, in blue, the risk, the added risk, if it's true that a particular participant in the study frequently consumes alcohol. So what's the problem with this? The problem is that you don't know what the risk factors are, and you don't know, therefore, which other risk factors to include. I've given you an illustration of what you might include, but you might include others. I haven't included smoking, for instance. And it matters which other ones you include because it can directly affect the conclusions of the risk factor that you're interested in. For instance, we know, based on lots of the studies, that obesity is important. If you if you don't include that in the model, but you include other factors which are somewhat correlated with obesity, then they're likely to come out as important, even if there's not a direct association there. So it's important to include the right predictors in your model in order to control for them statistically when assessing the evidence that the risk factor of interest really plays a part or not. And you could imagine investigating different models. You might include BMI or distance from the sea. You might look at whether they smoked or not and remove ethnicity. It may be that the risk factor of interest is not important at all, and in fact, that's the wrong model. So how do people usually go about addressing this problem? Well, there's a number of approaches. So one approach is to take those risk factors that you have some reason to believe are likely to be important and include all of them. Another approach is to take some kind of algorithm which will iteratively try to fit the model in such a way that there's an automated decision made at the end of it. But there's problems with both of these approaches, and the problems are that essentially the assumptions are not transparent, and it's not entirely clear what the results that you're going to get at the end of the process are. So ideally, what you'd like to do is systematically investigate all possible risk factors and then average over the likely models. And that's what we attempted to do using an approach known as Bayesian model averaging, which uses assumptions which prevent a particular symptom of an alternative approach, which is to throw every single variable at the model. You could ask me, why don't you just throw the kitchen sink at the problem and include all 1,000 variables that we extracted from the database? But if you take, for example, the outcome of mortality, the most severe outcome of COVID measured in this particular cohort, there were around about several hundred members of the UK Biobank who died due to COVID. But we have around about 1,000 variables that we're investigating. And so you've got many more variables there than you have observations. And so there's a great risk of overfitting the model. In fact, in a model such as this, you could even fool the computer or yourself into believing that you have perfect prediction for some combinations of these factors. And so what the Bayesian model averaging approach does is it applies assumptions that avoid overfitting. For example, it says perhaps that we expect no more than 10 risk factors to be included in the model. We just don't know which 10 it is. And that's not going to be a hard cutoff, but it's going to at least impose some kind of structure on the process. And ultimately, the quantity of interest, does drinking alcohol increase risk of COVID, is going to be interpreted by asking how often, over all of these different models that the computer investigates, how often is alcohol consumption included in the model or not? And that's known as the posterior inclusion probability. Let's go back to these results. This is a summary of the commonly cited headline risk factors for susceptibility to infection. And what you can see on the left column is the number of studies in the literature that made a headline out of these particular risk factors. And on the right-hand side is our probability expressed as a percentage that these are indeed important for susceptibility to COVID in this cohort. And you can see that there's a strong degree of congruence. After all, we're analysing the same data just using a different method. We're doing so at a different point in time, but that's probably a secondary consideration. And you can do the same for hospitalisation, and you can do the same for mortality. So what does all of this tell us? Well, the first thing that I noticed from this is that there's typically a lot of risk factors, and there's typically more risk factors. There appears to be more risk factors involved in susceptibility, hospitalisation, or mortality than individual papers typically headline. In other words, they tend to focus on one story, whereas the truth is rather less sparse. And in fact, a lot of the things which have been picked out by individual studies appear with very strong support to be important. Secondly, many of these potential risk factors appear to influence all the outcomes. So that would include deprivation, age, comorbidities, obesity, smoking, ethnicity, cardiovascular disease, alcohol intake, stroke. And then some of them influence particular outcomes. So some of them influence specifically hospitalization and death, and those would include lung disease, being male. Some of them appear specifically to influence infections such as exercise or mental health or diabetes. And so in a way, this mirrors what we're seeing at the genetic level, where there's some genetic risk factors which specifically appear to be involved in the process of infection, and some specifically in the process of severe disease given infection. Perhaps more interestingly, there appear to be risk factors which are relatively overlooked in the literature as it stands. So those would include, for instance, geography. Where you live in England seems to be very important, both for susceptibility, but also whether one is hospitalized. And then some of the widely reported potential risk factors, such as cholesterol, we find to perhaps not be so important. And so it might be that those are markers for something else that we've included, for example, general comorbidities, or perhaps cardiovascular disease, or even stroke. There's some limitations to the approach. Despite having 1,000 variables, there may still be some important ones which weren't measured and therefore not included. We've ignored this linear model approach. We've ignored interactions between the risk factors, and we've assumed additivity in the model. What we've not tapped into are individual medications that participants have used over the years, so that's excluded. And sometimes the results that we get, particularly for susceptibility, can be hard to interpret. Perhaps the biggest difference to the kinds of approaches which are typically pursued are that this Bayesian model averaging is extremely computationally intensive. We need to run each analysis 50 times to ensure that we've got convergence of the algorithms, and each one takes days of compute time. So you're talking at about 1,000 hours of computation time for each analysis, which is a lot. But on the other hand, I think the importance of these health outcomes probably merits it. Okay, so I hope that I've convinced you that data sciences has played an important role during the fight against the pandemic, and we're probably in a better position because of it. I think one thing that's clear is that many of the areas in which data science and indeed biomedical science were important, the impact was the result of pre-existing collaborations, research networks, or research programs. And so the ability to quickly and agilely pivot onto COVID research would not have been possible without those pre-existing resources in place. Okay, so if this has interested you, then I would encourage you to check out our program on data science, computing and maths. You can do so using the QR code, or you can Google Content Computing Maths. If you're interested in the artificial intelligence program, which I mentioned at the start, then you can follow this QR code, or you can Google Content AI Intro. And I'm excited to announce that some of the subjects that I've been talking about today are going to be covered in more detail with new weekly classes. These are 10-week programs for the next academic year, 2022 to 23. We've got a course on pandemic data science taught by Dr. Daniel O'Connor of the Oxford Vaccine Group, and an infectious disease modelling course taught by Dr. Emma Davis of the Oxford Big Data Institute. Both of those scientists were involved in the pandemic response. A personal interest of mine, data science and family history, will be taught by Dr. Olivia Robinson, a graduate of the University of Oxford, a postdoc at the Saxo Institute at the University of Copenhagen, and a professional ancestorian. And we have a new course on Turing machines and logic taught by Dr. Niccolo Salvatore of the London School of Economics. So thanks for listening, and I'll be happy if there's time to take any questions.
Okay, well, thanks very much indeed, Danny. That's really fascinating, and I'm sure there will be some questions. We've got some. David Graham has the chat questions, but let me start off with anyone in the lecture theatre. First of all, anyone would like to pose a question on what you've heard? Okay, Dan here first. Yeah, you first.
When you talk about alcohol, how do you measure, or how much do you trust the patients what they report?
Yeah, that's a good question.
It's always, we all eat more fruit and veg and do more exercise than we do, and we smoke less and drink less than we do.
Yeah, I'm sure that's the source of some error. The way that it's measured in UK Biobank is through different categories. So never drunk alcohol, do so frequently, and in between. And those would be based on surveys when the individuals were recruited. So not only might there be errors, but they might be out of date. So what that's likely to do is add noise to the analysis, and therefore perhaps make it look like the association of any risk factor is weaker than it truly is.
Yes, it's a very interesting talk and very interesting model you showed as well. So I was interested, you sort of mentioned interaction effects were ignored. And I was sort of wondering, is that a computational reason? Because you could imagine, like, some of your cholesterol, for example, high cholesterol and office-based job, for example, together they might have an effect that's not coming out in the model when you look at them individually.
Yeah, that's right. So that is the reason. It simply becomes with 1,000 variables, each of which might be included on its own, then you have a very large number, 2 to the power of 1,000. If you start including interactions, then it explodes even further. And from a computational aspect, there are some challenges. It does take a long time to run, and it would take possibly orders of magnitude longer once you start including interactions as well. But that's not to say that they're not likely to be important. I mean, anything that's known about biological systems down at a molecular level, it's known that there usually are important interactions. And one of the things that I haven't shown you is that we have applied other kinds of models to this, including from machine learning. The focus of machine learning is a bit different. The focus is really perhaps on prediction rather than inference as such. So it's more what I would put in the engineering box rather than the inference box. But we're still able to compare what the machine learners, many of which relax this assumption of additivity and therefore able to look at higher order interactions. We're able to compare what they think are important for prediction, at least, with the sorts of variables that we find from our inference approach. And often we find that they're quite different. And there's various reasons why that might be. But the short version is that we think that it's not asking exactly the same thing. Prediction is not the same as inference, and therefore it's telling us something different.
Great, at the back there.
This is all based on UK data, but you showed a map of surveillance data initiatives around the world, South Africa, Brazil, etc. So, I mean, any thoughts on whether some of these relationships like ethnicity would hold up if you look at an international data set?
Yes.
Whether that kind of analysis is done.
That's a good question. So the analysis has been done, and I believe that the same risk factors are seen, for example, in the US and elsewhere. So a lot of these, including ethnicity, are recapitulated between different countries. I don't know if I can tell you off the top of my head anything more specific than that, but I think it's certainly the case that non-white ethnicity is a risk factor in other countries as well as the UK. Although it's interesting that it appears that a part of that is social.
David,
yeah. Thanks, Danny. I mean, for your future research, what's the biggest thing you've learned, the most significant impact on your research career since December 2019? Because obviously you didn't plan.
Right.
To focus all this effort and energy and intelligence. So how's it affecting your future research ideas or?
Okay, good question. So it's scuppered several projects royally, and we're just trying to get those back up and running again. The collaboration with UK Biobank, which kind of led us in this direction, was already there, and so I suppose the hope is that, if anything, it will help by demonstrating the importance of infection as a trait, believe it or not, which was doubted by some in the UK, because of course infectious disease it's not at the top of the list of causes of early death in more developed countries. That's my hope. I also hope that perhaps these risk factor analyses, we might be able to roll them out more systematically across different diseases, so not focused just on COVID, but maybe looking at other ones like migraine, for instance, or completely different.
Okay, can I see any questions online? Okay, I know someone at the back, and then we'll take that one. Yeah, you're next.
Hi, thank you very much. It is, of course, highlighting a very challenging aspect of whether to try to look for commonality versus the individualism of a disease that we get to know from. To what extent then do you think looking at individual risk factors, let's say, for example, obesity, if you inspected in a more of a horizontal way, you get 2,000 patients who are obese, and one of them, not all of them, they will respond equally to the disease severity. This is at one hand. And on the other hand, to what extent then we can link the non-genetic risk factors with what we learned from the SNPs in the G1s. Okay.
So I think it's a question in two parts. The first is by necessity in the model, we're assuming that people, for instance, who have a level of obesity respond uniformly, or at least they have some average effect in terms of their change in risk. And so that's really, it's lumping people together in a way which isn't entirely realistic. And I accept that, but I also don't think there's probably an alternative to doing that, because fundamentally you need replication. So if you don't have replication, then you can't establish whether a signal is really believable or not, or is simply noise or some kind of artifact or idiosyncrasy. And the way in which in Data Science 1 achieves replication is by saying that certain things are equivalent to one another. And in your example, we're saying that in some sense, individuals with a level of obesity are equivalent to one another, and that by analyzing them as a group, we can make statements. And the fact that we do find signals suggests that there's some truth in that. The second part of your question was whether we can link the genetic and the non-genetic risk factors together. It's a very interesting question. It's one that I've not thought about so far.
Great. There were some questions online, David.
Yeah, I've got a question from Idalina saying, thank you very much for the lecture, very interesting. She was interested in the impact of geography that you mentioned. Could you say a bit more about this? Is it about access to healthcare facilities, or with regard to wealthier, poorer areas of the country, or something else?
That's a very good question. And okay, so what can I tell you about that? I can break it down a little bit into the sorts of risk factors that we found that I put under the heading of geography. So often we found that the part of the country where individuals were recruited from came up as significant, so significant differences. And I think this particularly applies for susceptibility to infection and for hospitalization. So perhaps we can understand that for more so in the early stages of the epidemic, when the risk of infection was very heterogeneous geographically. It's probably now that we are some way into the national epidemic, that's less the case. And then again, with hospitalization, it's not totally obvious that would explain it. So yes, it probably is measuring differences of the kind that you mentioned. Other risk factors that came out in that category included pollution levels. Is that because pollution per se is important, or because pollution is a marker of some kind of deprivation? That's possible. It also, one of the other things that came out was whether you live in the north or not. So northing was a significant risk factor, and although perhaps it wasn't quite as important, distance from the coast also came up. So I don't know. I would leave it to the geographers to decide exactly what the interpretation of all those things are.
Thanks. So any more online, David? No, that's it. Great. One down here. Yeah.
So I've just got as an ongoing thing for policy for future pandemics or something. I mean, is it possible to say how effective, say, shutting restaurants and other policies are, or is it still too early to do something like that?
Right. Yes. The Eat Out to Help Out scheme, for instance.
Yeah.
There have been analyses of that sort. There are different types. So there would be more in the surveillance and policy making examples of applications of data science, where one's trying to understand what particular measures have had and what their effect is, rather than looking at risk factors per se. But there are papers on this, and there are estimates of how much better or worse that particular scheme made things. And I don't think it made them better.
Okay. Down here.
Thank you. Yes, I'd like to know if there were any studies to try to quantify the effect of conspiracy theories and social networks on the anti-vaxxer movements and the rate of vaccinations in countries. For example, it's high in the UK or France, but it's not very high in Germany and in the US. Okay.
So the question is, how does one assess the effect of anti-vaxxer movements and such networks, social networks, on uptake of the vaccine? So what you would need to do, I'm not aware, but I wouldn't be surprised if there are research studies into this. But what you'd have to do is you'd have to have some way of measuring the local impact of those different phenomena, and then compare that to, for example, incidence of infection, or indeed disease through hospitalization or mortality. And you'd need to do so in quite a sophisticated way that takes into account the temporal and the spatial variation in those things at baseline throughout the pandemic. So it's a difficult question to address, but I'm sure there are people who are looking at it.
Great. Let's have one.
Representative are the people who are in the UK biobank of the UK population as a whole?
That's a good question. So they are not entirely representative. In various ways, they differ from the population as a whole. They tend to be more health aware. They're participants in research. There's a so-called healthy participant effect. So UK biobank participants, on average, were probably affected less in terms of their rates of infection and perhaps in terms of their adverse outcomes. They are also not a uniform survey of the UK. There's a central belt from sort of Liverpool to Hull, where there was a lot of the sampling done, and then dotted around the rest of the country. Most of the participants are from England, but there are also residents in Northern Ireland, Scotland, and Wales. So they're not representative, but I don't think that necessarily means that we can't take the conclusions of these sorts of analyses and say that they're probably relevant more generally.
Great. Okay, thanks so much. I think we'll leave it there. Just before we wrap up, I'd like to thank David Graham, who's our Research Development Manager, for doing all the work behind the scenes and preparing this with the technology and so on, and for being in charge of the live feed. So thank you very much indeed, David. And we'll be going for a drink in the common room, so do join us there. And finally, to thank Danny Wilson very much indeed for a fantastic talk.